All right. Hello, everybody. I'm going to talk about the statistical analysis of this, well, 16S rRNA datasets or something similar. There are several ways to do this. This is just one way to do it. It doesn't cover everything that you can do with the data, but it at least introduces the stuff that is currently available in Chipster. I used to work here at CSC with Chipster, for example. Nowadays I work at the Finnish tax administration. So, interesting choice, I guess. Now, the tools in Chipster, they are based on R. At least these ones are. And what we have here are the actual topics of the talk. So I'm going to talk a little bit of different kind of visualizations, either just by checking how the data looks like or doing some visual data analysis. But the main topic here is a statistical analysis for marker gene studies, or in this case just 16S. But you know what is R? Yeah, it's a statistical toolbox. And it's based on libraries, so packages that expand the functionality of R. So what are these here in these brackets? This is the package name in R, and that one here after 2 dots is the actual function name in that package that Chipster uses for doing those different kind of analysis. So you can do similar things in R, and this is just a little list of packages and functionalities that we are taking from R. All the slides are based on this Costello dataset, and we have only actually taken a small subset of them, just the stool samples. If you're more interested in that dataset, there's a paper on that, and there's also on Motor Wiki a walkthrough of the data analysis. Now, in Chipster, this is pretty simple. We only have one tool there that does all the analysis basically. Once you have the pre-processed dataset and the updated phenodata where you have filled in the group column, you can run the tool in Chipster. It would be nice to have a heatmap visualization, but it doesn't currently work in Chipster for metagenomics datasets. So what is in the tool then? First, a couple of words about rarefaction curve. So it's used for estimating species richness or abundance. Maybe richness in this case. It can be plotted as a function of the number of samples. For example, how many sequences you have sampled, or how many actual samples, for example, from different persons or animals or whatever. And then the number of species. Now, Chipster plots only the curves as a function of samples. If you use the R package, you can also plot the same curves as species richness per sample. So here's an example. This comes from the Chipster. So here on the x-axis you have the sites, or in this case samples from different persons, or the stool samples. And this is now the rarefaction of the number of species. Then we have two groups in this case. There are two curves, one red curve here, which is for group 2, and then this blue curve here on top of it, which is for group 1. And these colored areas around the solid lines, they are just confidence intervals. Now the idea from this is just to check whether you have sampled kind of both groups equally well. The curves should be pretty similar as they are in this case. For example, if these confidence intervals overlap, you know that the sampling efficiency in both of those groups is more or less the same. So it's just to check whether you have kind of taken enough samples. You can also draw it using R per sample. So here we have number of sequences in those samples, and then we have number of species on the y-axis here, and these are individual samples that are included in the dataset in this case. And the coloring. Yeah, it was on the first slide. Spec Akku from the vegan package. This is not currently available in chips, but chip in the software, but it should be pretty simple to add. So let's see when we get around to do that. But this is just to check the sampling efficiency. You can also draw a rank abundance curve. If you want to read more on that, there's a Wikipedia page. But it displays a relative species richness. Now, it also shows you the evenness. Sorry, I always want to point from the slide, but we are recording, so I have to point here. So if all the species here would be equally abundant in samples, there would be just a flat line through this plot. We have the species rank, so we calculate how many sequences you have per species, and then you rank them. The species that has the most sequences is plotted here on the top left, and then the next one, and so forth. And the abundance, or how many sequences you have per species, is here on the y-axis. Now, this is from all samples present in the dataset, but it is also split between the two groups. So here is for group 1, and here is for group 2. And now this grouping of samples comes from the phenodata column, or the group column that you filled in the phenodata. So everything that is in the group 1 is plotted here, and everything that is in group 2 is plotted here. For example, you can see that Clostridialis is the top most abundant group in this sample group 2, and it also comes on the top in the group 1. But you can check if everything is equally abundant, then this would just be a flat line. It typically looks something like this, not always. This is one way to visually see how different those two groups are. You can also draw it per sample if you like, and this is not currently available in Chipster either, but it is also very simple to add. So this is how it looks per sample. You see that samples look a bit different here, one line per sample, and then quite often there are a couple of deviating samples which have, let's say, small amount of sequences only, or something like that. Now, you can also, of course, visualize the dataset as a heat map. I originally programmed the tools in Chipster, so maybe I take the claim that it doesn't currently work for the metagenomics dataset, but this is how the heat map would look like if it worked. So we have samples here on the rows. There are sample names, so there's one row per sample, and then you have different groups of bacteria or whatever organisms you have in the samples here on the vertical axis. And if you check these, well, you know what is a heat map, do you? Who doesn't? Well, okay, what I'm talking about here then. But this is one rather nice way to visualize the total dataset too. If you have a very huge dataset, you can. Yeah, go ahead.
Just the counts, are they normalized or are they?
In this, they are not, but in here, they are actually scaled. So these are kind of relative abundances of these species inside every one of those samples. So this may be a more exact depiction of the actual dataset. one way which is nice to show here is that you can actually cluster the samples first using the k-means before you draw the heat map. It kind of makes the dataset smaller and allows you to see different kind of things from the heat map too. But we might add this in Chipster too. Okay, so those are the visual tools. Then onwards with more statistically oriented tools. You know what is an ordination analysis? Principal component analysis. Rings any bells? Okay. The idea is the same. If you have just, let's say, linear regression, you have just one response variable. We can, for example, predict the shoe size of the persons from the height of the person. So you have one response variable, which would be the shoe size, and then you have the explanatory variable, which would be the height of the person. So then you can fit in that dataset normal linear regression. And it tells you how much the shoe size changes when the height of the person changes, let's say by 1 centimeter. Now, we can also have several response variables. For example, in the case of multivariate analysis of variance, or AMOVA, you have several response variables that you want to model at the same time. Instead of just having the shoe size as the response variable, you could also have something like the color of the eyes and the IQ quotient or something like that. Now, AMOVA is good and well if you have normally distributed data. But for example, these data here, they actually count, so they are not necessarily normally distributed, so we can't use that for metagenomics datasets. There are other tools for that. One of those other tools is analysis of variance using distance matrices. We will come to it slightly later. But what people very often use is some kind of an ordination analysis. Now, the general idea is just to display a dataset where you have several dimensions, meaning that you have, for example, several explanatory variables. If you've taken samples from stool, like Costello, you could assume that things like gender, age, what people are eating, and so forth are having some effect on how the microbiota looks like. On top of that, what you measure from the stool samples is a total microbiota, so you have several species that you can identify from the samples. So there is one case where you actually have two kind of data sets, the data from the persons and data from the samples. And what ordination analysis is then used is you can try to kind of explain the things you see in the sample from the stool using the things that you have measured from the person. And this is what people use ordination analysis. So you have several dimensions, either response variables or the explanatory variables that you want to use in the analysis. This analysis basically just takes a species count table, which is now the table that you just generated, having the number of sequences in the cells. It also takes an environmental measurement matrix. In this case, in Chipster, it's called phenodata. And it basically just creates an image that allows testing for the significance of environmental factors, or in this case, the things you have measured from the persons, and how they affect species occurrence or frequency or abundance. There are two types of approaches to ordination. Here on the left is something which is called unconstrained ordination. And principal component analysis, or PCA, is one example of that. You can use that kind of an approach if you don't want to use the environmental measurements in the analysis, meaning those that were measured from the persons. If you just want to use the abundance data taken from the sequenced samples, then PCA might be something that you can do. Then there are constraint ordination analysis, which uses this environmental measurements in the analysis. And what people nowadays pretty much use are either CCA or RDA, redundancy analysis, or the correspondence analysis. The C here stands for the canonical, meaning that it is constrained by some other explanatory variables. Now, what you get out of it is a figure looking similar to this. Actually, let's skip to the next one. So you have two axes where you display all the data you have. So you have here, for example, the samples, which are the big colored dots, and these are colored by the group. If I now recall right, the red ones are from the group 1 and the blue ones are from the group 2. So these are the samples. Then you have the species or the bacteria that you have measured. Those are the gray dots. You see the gray dots? Good. Because not everybody does. So those gray dots there, they are those bacterial species. And then you have your explanatory variables taken from the phenodata in Chipster. And in this case, you just have the group column. It contains numbers 1 and 2 for groups 1 and 2. Now, the group value increases in the direction of the arrow. So you have group 1 somewhere here and group 2 somewhere here. And as you can see, the red ones are to the left in the image, and the group 2, the blue dots, they are to the right of the image. So this grouping actually explains something of the difference between the samples in our dataset. We also have a p-value for that effect. So the p-value for the group effect on how the bacterial biota looks like is 0.003. Now, using the normal rules of thumb, that would be statistically significant effect. So the grouping you have specified explains something of a difference between those two groups of samples. Usually you can do this with a couple of more explanatory variables. I've taken this from R because currently Chipster only allows you to use this group column to run the analysis. But you can add more information. For example, there's gender, the individual, and the day the sample was taken. So you see, for example, that the day is pointing downwards. So the more later days are actually here on the bottom, and the earlier days are here. We also have gender, which points to that direction. These are males here, females are into that direction, and then you have individuals pointing to that direction. Individuals were just numbered by a running number. So you have individuals with a higher running number in this direction. You can basically try to figure out how those dots here, these samples, fall along these explanatory variables here. What you see, for example, if you plot every one of those dots on here, they should be perpendicularly kind of projected, so you can see in which order they fall. This is actually pretty hard to do on the computer screen, but anyway. You see probably pretty easily that gender, for example, tells those dots apart, because if you think along this line here, the blue ones are here and the red ones are here. Also, day tells the samples apart, because the earlier days are here and the later days are here, and the red ones actually are taken on the later days more than the blue ones, for example. And then you have this p-value for the group column again, which is now actually the gender here. So in Chipster you only have possibility for testing one of those variables that you have measured from the individuals at a time, but it can be pretty easily done, for example, in R, and we can probably also program it into Chipster too to support several explanatory variables in the same analysis. This is a pretty standard way to do the analysis nowadays. Now, I just added a couple of interpretation slides, maybe not go into details on those, just note that reading those images is not always as simple as it would look like. These are available in the material, so probably you can check them there. Just to note that there are two different kind of ways to draw the image. There's type 1 and type 2. Whatever you check, they always tell you about type 1 and type 2 scaling, which is how you read the image. Now Chipster uses type 3 scaling, which is a kind of a odd species, because it's kind of a mix between these two type of scaling, so ways to read the image. Any questions this far? Okay.
Can I ask something? So,
Sure.
So what's the difference? Why couldn't I just use the principal component analysis for this? I mean, it's to separate the groups, and or this RDA gives me also the effect of one this feature in the sorry, maybe I don't understand this.
Yeah, well, the basic difference is that this is PCA here,
Yeah.
or here. You just have the raw data, or sites times species matrix. So you have this count table that you already saw in Chipster, and this is what you use for analysis, basically. It gives you this kind of a similar looking ordination biplot.
Yeah.
Whereas in this constraint, or RDA thingy, you also take into account the explanatory variables, so variables that you measured from these sites, whatever those sites are. They could be individual persons, plots in the forest, or whatever. And this is similar to, let's say, regression analysis, where you have some kind of explanatory variables that you can take into account in the analysis, whereas this PCA doesn't take these explanatory variables into account at all.
Okay.
I have another question.
Yeah.
Is the RDA the sum of the different variables? When you have the percentage, for instance, of it?
Yeah, you mean these percentages here? Yeah, I didn't want to go into that, but it's kind of a part of the explained variance, basically.
So the more variables we have, the more we can explain.
Sure. You typically only plot 2 x's in the plot. Well, you can put there 3, but it's harder to read because you have to rotate it.
And then after the following slide, you show p-value and you say it's associated to gender, but in that case, can you find out what are the p-value for those arguments that you are like both the
You mean also for these ones?
say this p-value is showing gender, right?
Yeah.
And what about the other one? Can you find out what it is? Or?
Yeah, in the analysis you can.
Yeah.
But in Chipster, it's currently only implemented so that you can only use this group column.
And how many are you using R?
Mm-hmm.
How many type of variables do you use to put together?
Where basically you can put as many explanatory variables as you like, but it's typically said that you shouldn't use overly many, because then it becomes more and more like the PCA, because it's not then that constrained anymore. So a few, several, but not tons.
Just thinking about the bacterial data, because there's extreme the three differences between the different columns, like in the abundance of species, that would it be better to use some like this model-based multivariate analysis that can be a bond and the Gordon approach?
Yeah, you could. The basic problem with the ordination analysis typically is that you have many zeros in the matrix. Now, in Chipster, the dataset has been Hellinger transformed, which should kind of at least partly take into account that problem. So, well, let's say that's the state of the art, but that's for the ordination analysis almost as state of the art as it gets. But certainly you can use something different too. I haven't actually seen any papers comparing those methods directly in so much detail, so I couldn't comment which is better, but all right, a couple of things about statistical analysis still. They are all put in Chipster into the same tool, but sorry, it partitions the diversity of the samples into alpha, beta, and gamma diversity, which are listed here. I'm not going to talk about that in any more details, but just to let you know that you can see how the diversity can be kind of partitioned into three different parts. This is pretty often seen in papers. So the tool does that for you too. But if you just want to compare the groups, there are several ways you can do that. The one we already saw, that's the ordination analysis. There are also other statistical tools that you can run. One is the permutational multivariate analysis of variance using distance matrices. Do I have to read these? Well, there are three different tools you have. And basically, they are testing exactly the same thing. Do the groups that you have specified in Chipster in group column or in general in some other variable, are they somehow different? They use slightly different methodology for calculating, for example, the p-values and the actual tests, which explains that they might give slightly different results. But anyway, there's this permutational test, multivariate homogeneity test, and the analysis of molecular variance. And Chipster lists all of these. And there's also indicator species approach that you can by default use for checking what are the taxa, the species that differentiate between the groups in the best possible way, or which are the species that you can use indicators for certain kind of an environment. So here are variance analysis. Just very shortly, the background. If you want to read more on the test, here's the first publication, but it uses a similar kind of approach than the normal variance analysis, but it's based on distance matrix and gives out a p-value. Now, here's how it looks in the output. This is taken directly from R. This is what you're testing. DD here is just your environmental species size matrix, and then you have the grouping of the samples here. Then you have the variance partitioned here, where you can see that the total, this is the total variance. Then you have the part of the variance that grouping of the samples explains. This is a mean standard deviation for the grouping, which gives you the actual statistical test value that you can then turn into a p-value, which is here. So this test gives now a result that the grouping explains part of the differences between the samples, but the p-value is not statistically significant. because it's larger than 0.05. We can use a different type of an analysis to do exactly the same thing. This is not, again, available in Chipster, but this is especially good if you have crossed or nested factors. So depending on your experimental setup, this might be the test you want to use if you have crossed or nested factors in your test. These datasets here might or might not have such experimental setups. Now, it again gives you out a p-value. In this case, again, it gives you a p-value for the group, which is pretty similar to the earlier one. The difference is not statistically significant. It also by default gives you result from the third statistical test, which can be described as a multivariate analog of the Levin's test for variances. And there's again the publication. And it again spits out a p-value here, which is even larger than from the two previous tests, telling you that the difference is not statistically significant. So these are the three statistical tests on top of the ordination analysis. Then you can also check what are the parts of the diversity, and this is the publication that you can check if you like, but it basically reports you the different parts of the diversity. The basic idea is that a sample or a unit that has many unique species has a higher contribution to the diversity than the samples that have more equal diversity. Now this is how it looks like. So this is a diversity partitioned into gamma, beta and alpha diversity here in these columns, and then you have different kind of taxa here on the rows. So this just gives you a general idea about how the diversity looks like on the basis of the dataset. You can also run indicator species approach, which is described in the paper here, and gives out basically the indicator value of the species. And now it's run against the grouping that you have specified in the phenodata. It also gives you p-values for those indicators. Now, let's jump here. This is how it looks like in Chipster output. So this is from the indicator species approach. And the idea is just to see which species are associated with different groups in principle. So in this case we just have two groups. We have p-values for those taxa. And here, for example, this cyanobacteria, which is pretty interesting because these are stool samples, there are cyanobacteria present. Well, it happens. But it seems to be that this cyanobacteria group is one of the indicator species in this dataset. Its p-value is less than 0.05, so it seems to be able to differentiate between the groups. And if you look at how the indicator values look like, here's the cyanobacteria group here. It seems to be associated with group 1 and has a very low association with a group 2. So if you find cyanobacteria in the stool sample, you can then maybe try to, let's say, predict that it belongs to group 1, for example. There's another indicator species approach available in Chipster 2, which is described again in that paper in more details. It's interesting that everybody still cites this paper as 2008 in press, well, I don't want to publish in that journal. Takes forever to get the paper out. I'll just jump to the next slide, which basically gives you this ISA score. And the do I have the nope. I was just checking out for the cyanobacteria, but I didn't put it in here. These are slightly more esoteric to interpret, and I tend to like the previous indicator species approach more. But there's just a different approach for making a kind of an indicator species analysis. That paper is actually rather good for explaining what those numbers exactly are. Here's how it's basically calculated. It's just slightly hard to see from the matrix. And in Chipster it looks slightly different, but still the similar kind of numbers coming out from that too. Now, we have gone through several topics as statistical analysis, how you can compare the basically diversity and abundance between groups, either by ordination analysis or statistical analysis, and then a couple of things about visualization. The next thing would then, I guess, be to see how this is done in Chipster. This would rather be so simple that we can just jump to the exercise 15, I guess. Of course, if you have any questions, just shout. Or twist and shout, it's up to you.